Students, welcome back to second year botany class. So till now we have seen the two LQs from the 13th chapter, that is the strategies for enhancement of food production. That is the plant breeding process also we have seen, as well as the tissue culture techniques. Okay, so in that, out of that, we can expect one. This year only they have given the one of the LQ from the 14th chapter, that is the sewage treatment. So they have considered that unit as the capacity to give 8 marks question. So most of them, why the backlog is there in the botany means because of this reason only I am guessing. Okay, so just to come out of that strategy, that from this chapter only this LAQ unit you have to consider over here. Okay, once we have taken out the unit means we will take out the chapters that are present in that unit. So 13th and the 14th all together one unit. From that unit, we can guess we can have one LAQ and the one VSAQ or two VSAQs. Okay, that's their wish and will. Now one SAQ or one VSAQ also they can give, and another VSAQ they can take it from the first unit also. That I cannot guess. But we can make sure that at least two VSAQs from these two units, that is 1 VSAQ from the 1 chapter that is present in the last unit of the second year syllabus for us. So here we will see the 14th chapter sewage treatment. So in this, no diagram, nothing. Okay, only the flow chart will be there. That flow chart also, according to the theory, what we learned, same if you draw the flow chart, that's easiest one. Next comes only the words, what we have to use over there makes the much more impact on the answer. And the evaluating person also searches those words. So pick up those words and make sure that the word has to be used in the specific sentence. Then you will get 8 out of 8 marks from this answer itself. So that is the first one is the, in this chapter, only one LAQ is there for us, that is the sewage treatment. Yeah, so sewage treatment, explain, describe about the sewage treatment. Like that also they can ask, and whatever that means the how we will do the sewage treatment in the urban cities, that also they can ask. Whatever may be the question, but the answer should have these points in it. So first of all, what is meant by the sewage treatment? That is, in this also we have to write the meaning for the sewage word first, then the meaning for the sewage treatment. So 2 sentences we have to maintain over here. Okay. Yeah. So the first one is the what is meant by sewage. Sewage means the wastewater that is produced in the urban, this is from the urban cities, and that is dumped into the river sources. That is the sewage treatment. That is the sewage water. So the wastewater that is generated from the urban areas, mostly having the human excretia. Okay, so that is called as the sewage water. This sewage water needs to be treated first. That means it has to be cleaned. So the sewage water now which is going to be cleaned by different steps is called as the sewage water treatment. That is the purification of the sewage water also we can say. Otherwise, if you don't remember this word, you can say cleaning the sewage water before entry into the sewage water into the river sources also you can say. Okay, so this sewage treatment is considering having the 2 steps. There is a primary treatment and the secondary treatment in them. So first one is the primary treatment. Next slide. So in the primary treatment, what we will do? This is nothing but the everyone can do without any knowledge also the person can do the primary treatment. So in the primary treatment, first of all, whatever the sewage water that was collected, we will dump in a container, in a tank. So that is and in that we will remove the we will remove the things that are present as the floating debris. Okay, floating materials, that is the leaves, branches, or plastic bags or anything. Everything we can easily remove by the physical filtration process. That also has to be done in a stepwise manner for many times. That is called as the sequential filtration. And after that, once we have separated we have filtered the floating parts from the sewage water, then the water that is present will be having the solid particles, that is the pebbles, otherwise the soil molecules and everything will be settled down at the bottom of the container. So that is called as the sedimentation. So this one can be removed with the sending out this water into the another container. So the settled part at the bottom of the container can be left out like this, and we can remove it afterwards. And the third one comes with once the pebbles, otherwise the solid particles which were settled down at the bottom, that is called as the primary sludge. And this primary sludge, what it is having? It is having the soil particles and the pebbles in it. Pebbles means small stones to the big stones. Okay, those are called as the pebbles in the primary sludge. That is the waste one, what we have to remove. And the one that is present, the water that is present above this primary sludge is called as the effluent. And that also, this occurs in the primary treatment, that is in the first step of the sewage treatment. So we can call it as a primary effluent. Okay, so this primary effluent is also called as the supernatant. We can use any word. Effluent is easy for you, then you can use only the effluent word. Okay, next we will go with the secondary treatment. Now the primary effluent was sent into the second container for the secondary treatment. So in the secondary treatment, we need to have 3 containers, and in that one first container, the primary effluent is going to be coming from the primary treatment, and that one has to be stirred mechanically. That means with the long stick, otherwise a rod, we have to stir it, and that with that all the organic matter that is present in the primary effluent is going to be mixed up very properly, very easily, so that entire air will be rotated in the container also. Top to bottom air will be rotated. So that is the aeration also we can call. And in that, once we have stirred that entire effluent, primary effluent with the stirrer, otherwise with the mechanic rods, otherwise the stick or anything, then the air will be rotated, oxygen that will be rotated. So with this helps out for the formation of the aerobic microorganisms. And these microorganisms will start their growth vigorously by the presence of the oxygen in them, and they will make the flocks formation. Flocks means bacteria and the fungal organisms together makes the colonies on the surface of the water. Those are called as the flocks. And these flocks, these flocks has to be removed. So we can easily remove these flocks with the filters, microfilters, and then we have to transfer this water into the second tank, where only the microorganisms need to grow very fast. So in that container, what we will see, the oxygen availability we will see. So in that, what is meant by BOD? BOD means biological oxygen demand. If once the aeration was more and the flocks were formed, these flocks will consume the oxygen and start their growth. How they will do the growth? By utilizing the organic matter that is present in the sewage water. They utilizing means they will consume it and they will grow vigorously on the sewage water surface and makes the flocks. So with this, oxygen levels will decrease in the second tank of the secondary treatment. So with that, what we can say, if the oxygen levels are decreased, then the organic matter is going to be utilized properly by the microorganisms. then the organic matter quantity will be less. Then the water is somewhat pure now without the organic matter. So it can be, we can easily remove the microorganisms. So that is the, so 1 meter will be present. So with that meter, we will check out the oxygen quantity in the water. So if the oxygen quantity is less, that is the levels are less, then we can say all the entire organic matter was consumed properly by these microorganisms to form the flocks. Then we can transfer this one into the third container, where we will, in between that, we will use the microfilter so that all the flocks can be removed and only the water will be sent into the third container from this in the secondary treatment. Now, these flocks will be sedimented. That means we will take out with the filters, and that is called as the activated sludge. And once they will settle down at the bottom of the container during the secondary treatment, that is called as the sludge. And because of the heavy nature, they will settle down at the bottom, and that can be easily removed. How we are transferring from this container to the last container now? So like that, we are transferring only the water, not this microorganisms at all. So that is the activated sludge. So this activated sludge that is present at the bottom of that container can be reutilized, that is inoculated on the primary container, primary treatment, where we will go with the treating the, that means removing the debris, otherwise the floating particles with the filtration process, everything what we did in the primary treatment, that again we can go with that process. So there the treatment will be very fast if we use the activated sludge in the primary treatment. Now this small amount of the activated sludge will be inoculated. That means once we kept on the aeration tank, otherwise in the primary treatment, that is called as the inoculation. Okay, making them to grow forcibly in that medium, that is called as the inoculum. Okay, and now this water what we have transferred into the last container in the secondary treatment, where oxygen levels are very less. Whenever the oxygen levels are less, then the anaerobic respiration starts for the organisms, where they will consume, otherwise they will use, otherwise they will completely take out the bacteria which were formed flocks in the water. So like that, we can take out the bacteria and the fungi by the anaerobic organisms, and they will keep on digesting the heaviest particles which are present still in the water also. Like that, we can make the water clear from the organic and the inorganic substances with the anaerobic organisms. So by this process, what we are getting, this is the last step in the secondary treatment. After this, the 3 different gases are going to be produced. That is the carbon dioxide, methane, and the hydrogen sulfide. Okay, so 3 different types of gases are released, and all these gases are released into the outside atmosphere from the container, so that the water will be free from the now organic and the inorganic particles. Now this water can be easily dumped, otherwise transferred to the river sources, otherwise the natural water sources, so that no further water pollution can be seen. All the aquatic organisms can survive very easily, and the soil water, that means the groundwater, also not polluted with this type of water that is dumped into the river sources. That is the completion of the sewage treatment. And the same thing, the government also have utilized, otherwise taken the chances to clean the river, natural river sources for us. That is the last slide. That is the Ganga action plans and the Yamuna action plans, because these are the major freshwater resources in India, where all these rivers are going to be spoiled by the wastages that are dumped from the factories. So that has to be cleared. So again here, the Forest Department and the Ministry of Environmental together taken an action to clear the wastages that are dumped from the factories of all different types of factories, dumped the water, so that the complete river is on danger right now, the Ganga river. because of the impurities that the factories were sending into the river sources. So that is the Ganga action plan and the Yamuna action plan. But the implementation is a very difficult task, but they are going to try to do this work. So with this, we have finished the third LAQ from the unit. Now we will start the easiest SAQs from the syllabus for us. Okay. So yeah, there only. Oh, thank you. Black only. Okay. So now we have finished the 3 LAQs from this unit itself. That is the plant breeding process, tissue culture, and the sewage treatment. Out of these 3, 1 will be coming in the final exams for the supply students. Okay. So now we will go with the 1 LAQ. Once we have prepared, that's sufficient for this time movement. And once we have finished the most important SAQs and the VSAQs, again, I will say the second LAQ also, so that at least a half, otherwise 3/4 of the LAQ you have written. That means you will get nearly 5 marks. It's not a big deal for us to get the 5 marks if the supply students are there. So that is the most important one, somewhat bigger, and you need to have the confidence on me, first of all, to go through that LAQ, and the interest need to be retained. So first of all, we'll go with the SAQ part right now. Okay. So in this, the SAQs, the most important SAQs are those are coming from the bacteria and the viral organisms. From these 2 chapters, 1 SAQ we will get. Okay. So 1 SAQ and 1 VSAQ from the bacteria and the virus. From these 2 chapters, we will get 1 4 marks question and 1 2 marks question. So from which chapter they will give which question, we cannot say. Mostly we will say from the bacteria, but sometimes they may give from the virus also. So we cannot say here from which chapter we will get the SAQ or else the VSAQ. So better to learn these 2 chapters, SAQs and the VSAQs, in a proper way, so that we can score 6 marks. So 8 over there, here 6 means 14. And if we prepare with another 2 chapters, that means 8 marks, then easily we can get the pass marks, isn't it? So 8 plus here 6. So 14 plus 1 4 marks question and 1 more 4 marks question. That means 8. So here 14 plus 8, 22. And once we have prepared with the 4 marks, means at least 2 VSAQs we can answer. That means 4 marks again, 26. If any spelling mistakes or anything happened, then also easily we can score 21. It's a very damn easy task, isn't it? So how we are preparing, first of all, whatever may be the question they are giving, at least if you know the concept, if you have written on your own also, you will get the marks in the sewage treatment. But some people have, by seeing that question only, you have lost your memory and you have failed. That's the reason the students who have failed in this subject, second year botany, nothing else, not that much big deal to get 21 marks in my subject. First of all, you should be strong enough mentally so that whatever may be the question comes, you have to think from which chapter it is coming. If you know about the chapter, at least to say few words about that concept. In that, at least one sentence will be right. That means they can give you 1 mark now. So why you are wasting the entire marks and your brain itself? So use in the technically. Now let's say now everyone knows about the usage of the smartphone in different ways. So same brain use and getting the marks over here. Don't become a dumb over there by seeing the question paper. Okay. So how to get the 21 marks is there? Very easiest task. So get this, try this first. So to get this, we have to prepare for 6 marks first of all. So 3 SAQs and 4 VSAQs and 1 LAQ pass, isn't it? So we will go according to this strategy only now, because 1 LAQ was over right now. What we dealt in these 3 classes, 1 LAQ we have finished. Now your work is to revise and revise and revise. It's not my work right now. My work is to give the easy tips for you to remember and recollect. Recollecting and writing part should be done. That is, finishing work should be done by you. Just like how MS Dhoni will do in the T20 matches. Finisher, good finisher, what will say? Same thing we have to do. That you are the finishers over here, not we. We are guiding. So you have to do this work. Now prepare well with that 1 LAQ. Now we will start with the most important and the easiest SAQs. And along with that, the VSAQs also coming. I'll say for that also along with them. So the first one is the conjugation process in bacteria. So this is in the 7th chapter, that is a bacterial chapter. This is the SAQ. And here everyone will do the mistake is not writing the answer properly and not at all drawing the diagrams for this. Both are very important ones. So conjugation in the bacteria. So what is the question? Describe about the conjugation process. So in this, what we have to write? Conjugation as a type of the sexual reproduction that occurs in the bacteria. That is the first sentence. And the next one will be, this was the first given by Liederberg and Tatum. Liederberg and Tatum in E. coli. In E. coli bacteria. This much you have to write. Now comes with the third step, third point in this. That is how the conjugation occurs. Okay. So I will draw the diagram and I will explain. After that, I'll say the answer, what you have to write. Okay. So first of all, see how the conjugation occurs. Conjugation occurs in between 2 rod-shaped bacteria. In that, one bacteria should have the sex pili and the another bacteria may have, may not have. It does not matter for the conjugation process. But one bacteria should have the sex pili in it. Okay, this is the one bacteria what we have to draw first, which is having this one is the plasmid DNA, and this is the genophore. Genophore. And this is the sex pili. Now, in the second bacteria, is having the genophore, but not the plasmid and no sex pili. So, the bacteria which is having the plasmid DNA in it is called as the F plus bacteria, and the bacteria which does not have the plasmid is called as the F minus bacteria. Okay, and in this, what we have to say, what is meant by conjugation? Now I am saying the definition. Conjugation means transfer of genetic material from one bacteria to another bacteria through direct contact is called as the conjugation. So how to go with the direct conjugation? With this sex pili that is going to be attached to this F minus bacteria, then transfer of the genetic material. So which type of genetic material need to be transferred? Now the double-stranded plasmid DNA. Okay. So the genophore that is present, it should be present in the center of the bacteria. And the bacteria, it's having the black colored one is the cell wall, and this is the plasma membrane. Okay. Now comes with the second diagram. What happens after this? The F plus bacteria now elongates its sex pili and holds the F minus bacteria So this is the F plus cell and this is the F minus cell. So now what happens? The F plus cell makes the sex pili to grow and holds the F minus cell so that it comes nearer to the F plus cell. Now so that the conjugation can be done very successfully. So now the sex pili which is attaching to the F minus cell, that means holding the F minus cell. Now this is called as the conjugation tube. Conjugation tube. The sex pili now looks like a tube-like structure which is joining the two F plus and the F minus bacteria. Now this is called as the conjugation tube. Okay. Now what happens over here after this? Once the replication process starts in the bacteria, in the F plus bacteria, replication process will start. This is the genophore. This is the genophore. Okay. Now what happens? This is the F plus. This is the F minus. So what happens now? The plasmid DNA starts replicating itself to make a duplicate copy surrounding it. So I have not drawn with the same color so that you will get confused now. So I have drawn with the another color only. Okay. So the plasmid DNA gets replicated, and this replicated plasmid DNA now enters into the F minus cell through the conjugation tube. So that when it has to enter through the conjugation tube, the plasma membrane will be opened at that place only where the conjugation tube was there. There, the plasma membrane splits and gives the free entry, makes a free entry for this replicated plasmid DNA. Okay, so this is the replicated plasmid DNA. Okay, like this, the replicated plasmid DNA passes through the conjugation tube into the F minus cell and settles at the one corner of the bacteria, rod-shaped bacteria. Now, immediately what happens? The plasma membrane starts healing. It starts healing over there and makes a continuous membrane. So that it stops the passage of the once the plasma membrane heals that area, immediately it stops the passage of the replicated DNA through the conjugation tube, and whatever the amount of the replicated DNA enters into the F minus cell will be retained over there, and it will make its own copy here. Okay, like that, the F minus cell now turns into F plus cell. That is the conjugation process. Now, after this, what happens? The replicated DNA settles down at one corner of the bacteria and makes its own copy and makes the double-stranded plasmid DNA. Because DNA is the one genetic material which is having a capacity to regenerate itself. That means it can make its complete copy if it is having a small portion of the DNA. So that is the one what we are seeing over here also. Okay, so the newly formed replicated DNA, whatever the amount has been entered into the F minus cell, now makes its own copy, complete copy of the plasmid DNA was made. Now the F minus cell turns into the F plus cell. Like that, now by the end of this conjugate, once the plasmid DNA was formed in the F minus cell, now the F minus cell called as the F plus cell, and the conjugation tube breaks at the center and two bacteria were separated. So how this conjugation tube was broken down? By the formation of the septal wall. By the formation of the septal wall in it. Then the conjugation tube breaks at the septal wall and two bacteria were separated, and both are now having a capacity to go with the conjugation process of the same sexual type of reproduction with the other bacteria which does not have the plasmid DNA in it. Like that, in the bacteria conjugation process occurs. That is a type of the sexual reproduction. Okay, so now I will say what you have to write in the answer. Okay, so conjugation. What is meant by conjugation? Conjugation is a type of the sexual type of reproduction that occurs in the bacteria, and the conjugation means transfer of the genetic material from one bacteria to another bacteria through physical contact. That is the conjugation. So this was the first observed by Liederberg and Tatum in E. coli. So now we have to explain about this conjugation process. Conjugation process starts from the bacteria which is having plasmid DNA in it and the sex pili. And once the bacteria which is having the plasmid DNA and the sex pili, those who are starting this conjugation process are called as the F plus cells. And another cell which does not have the plasmid DNA but participating in the conjugation are called as the F minus cells. And now the conjugation, the sex pili elongates and holds the F minus cell. Now this sex pili is called as the conjugation tube. Through this, the plasmid DNA can be transferred into the F minus cells. After this, the plasmid DNA will start duplicating it. So that is the replication of plasmid DNA occurs. Here, only two words. In that, the specific word is the replication. Remember, it's not the duplication. Duplication is the low standard English, what we can say. Once the process, once we have to say for this process, conjugation, we have to say it as a replication, replicated DNA. So now the plasmid DNA undergoes replication process. Through that, another copy of the plasmid DNA is going to be formed surrounding it, which passes through the conjugation tube into the F minus cell. So after some time, the plasma membrane was closing this opening which was there at the conjugation tube, so that further transfer of the newly formed plasmid DNA was interrupted, otherwise was stopped. Immediately, whatever the amount of the plasmid DNA enters in the F minus cell moves to one corner of the bacteria. There it starts making, starts replication to make the complete copy of the plasmid DNA in that cell. Okay. Now, what happens in the conjugation tube? A thin septal wall is going to be formed in the exactly in the center of the conjugation tube, so that the two bacteria can be separated, and both bacteria will have sex pili right now. Both are having the sex pili. That is the intention of this conjugation process. Okay. And the last step is once the plasmid was gained its original shape in the F minus cell, now that cell also called as the F plus cell. And the conjugation tube breaks at the septal point, at the septal wall, and the conjugation tube broken down. That means two bacteria were separated, and each and every bacteria now having its own plasmid DNA. So that is called as the F plus cell. And this bacteria can go with the conjugation process with another bacteria which does not have the plasmid DNA in it. Like this, the conjugation process is a continuous process that occurs in the bacterial cells. Okay, these 4 diagrams you have to draw, and you have to mention the scientist and in which organism they have observed this process and what is meant by conjugation we have to write. And the labeling is also very, very important. And in this, ultimately what happens? F minus cell becomes F plus cell. This much we have, this F minus F plus cell indication should be there. Conjugation tube labeling and the genophore labeling and the plasmid DNA, replicated plasmid DNA labeling is important. And the sex pili now for both the cells after the division. So like this, we have to draw, we have to give the labeling, and we have to explain. So if you know the diagram, theory will come. If you know the theory, you can draw the diagram. So mutually benefited. Okay, we are living like a symbiotic. Okay, if we know the diagram, we can give the answer. If we know the answer, we can draw the diagram. So diagram is mandatory in this answer. Never forget that. If whatever the material you are following, I don't know, but diagram is mandatory. Okay, without diagram, answer theory does not have any value. If a diagram is there, then your theory can be considered as you have written on your own. Otherwise, it can be considered as you have copied somewhere else. Yeah, in my subject, this is the scenario what we will follow. So make sure that you have to draw the diagram at any cost. Okay, and we will see the next, in the next class, we will see the next SAQs, most important. Tomorrow, we will see nearly 4 to 5 SAQs. Okay, so that it will be easy for you to cope up with the supply exams and revise whatever the SAQs I am going to say, discuss over here, because chapter wise we will go. So tomorrow might be, I'm expecting that I can wind up 2 chapters SAQs, that is the bacteria and the viral organism SAQs, so that it will be easy for you to wind up 1 SAQ and 1 VSAQ. Okay, so stay tuned on the same time. Tomorrow we'll meet again with the 4 SAQs from the bacteria and the viral chapter. Thank you.